Hello, my Year 12s. Welcome back. It's Mr. D here. Let's go through the first PowerPoint you'll be doing at school. Okay, so just a quick recap of what we did in our Head Start week. Okay, so pretty much we looked at a bunch of different things, recapped Year 11 biology, and then went through the protein secretory pathway and the different things we have here. So as you can see, we have the PSP here. All right. So it goes all the way from the nucleus all the way outside of the cell in this example. So pretty much we're looking at the ribosomes. Well, really we're looking at the nucleus first, and the DNA inside that. Then how that's turned into messenger RNA through the process of transcription, all right, which we're going to look at later. Then that leaves the nucleus through the nuclear pores, and it will go into the rough endoplasmic reticulum, and it will find the ribosomes either on the rough endoplasmic reticulum or the ribosomes that are obviously out there in the cell cytosol. Okay. After that, it leaves the cell. Not the cell. It leaves the rough endoplasmic reticulum through a transport vesicle. And as you can see, the protein now enters the Golgi apparatus, where it then will get changed, modified, added to, anything of that nature, and it will leave through exocytosis. Okay, that's our last step. So exiting the cell in once again another vesicle. Cool. Alrighty, so essentially that's just covering what I just said. So proteins produced at the ribosomes from messenger RNA in the rough endoplasmic reticulum, as well as the ribosomes that are in the cell cytosol, transported via transport vesicles to the Golgi, modified more, changed, added to, and then exocytosis to leave the cell. Cool. And this is essentially just showing exocytosis. All right, so you have that vesicle there. All right. And the material inside will be excreted as the phospholipid bilayers join. So you can see the vesicle has a phospholipid bilayer, and you can see the cell membrane has a phospholipid bilayer. As they join together, it releases the contents, which are going to be proteins in this case. Cool. All right, so we'll get on to the start of it. So nucleic acids and nucleotides. So yeah, this is your first class for Year 12 biology. Welcome. Cool. So again, these are our key knowledge dot points from the study design. So we're just looking at this first dot point today. All right. So we also learnt about biomacromolecules within our Head Start week, as well as the monomers of different biomacromolecules. So as you can see here, you have your lipid first. All right. Could be called a triglyceride, classic fats. So that's your biomacromolecule, big chain of essentially either glycerol or fatty acids together. Okay, because they are the monomers, they are the single units. All right, again, looking at a polysaccharide, for example, starch here, so that's your carbohydrates. All right, that's your polymer. All right, so you can see there's multiple just sets of these monosaccharides. All right, so the individual units there. You can see one here, mono meaning one, saccharide meaning sugar, poly meaning lots, saccharide meaning sugar. All right, we also looked at proteins. All right, so amino acid being the smallest unit, so that's our monomer. And then we have our polypeptide, also known as our protein. So you can see it's just a massive amount of these amino acids, and they're all folded and bent into shape to produce a certain protein. Remembering our different types, different levels of the shape of the protein. And lastly, we're looking at nucleic acids in this class. So we're going to look at the nucleotide itself. All right. So that's your monomer. And then you can see, bang, all these are just one nucleotide stacked on top of each other. Boom, boom, all connected. Okay. And that makes your nucleic acid. In this case, the example here, this is RNA. How can we tell? Because it's only a single strand. Okay. Whereas you would have another nucleotide connected here. in an anti-sense way. So in other words, it's upside down compared to this one, or opposite. So if you had all of these connected here, 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 then it would be DNA. Okay? Cool. Again, this is the same image, just represented in a different way. Obviously, this is DNA this time because it has that double helix backbone instead of the singular. All right, so a large macromolecule is our DNA. There's heaps and heaps of atoms. All right, we looked at the nucleotide, which are the small segments. So you could just say it's as simple as something like this light blue area, this dark blue area, and our nitrogenous base. So thymine here, so that's one nucleotide. All right, light area, dark area, nitrogenous base. Another, there would be another here in theory, another here, and then this one, all right, which is obviously red, C, cytosine. Simple. All right, sugar phosphate backbone, that's what we call that, because this is essentially the sugar, which we're going to look at the specific sugar and the phosphate. Okay. How they bonded. So we know that the adenine and thymine here are bonded with H bonds, hydrogen bonds, cytosine and guanine. All right, and something to note is that they are quite weak, so they're easily split. All right. And of course, one pairing is weaker than another, which will come into biology in a few classes' time. Okay. And each side runs anti-parallel. All right, so what does that mean? It means essentially it's upside down. So you can see arrows representing that here. So arrow going down, and then the arrow's going up this time. All right, so they're running anti-parallel. And of course, they have the complementary base pairs, always. So you'll never see a cytosine with anything else but a guanine. All right, unless, of course, there's a mutation or something like that. Or something goes wrong, wrong repairing, which can happen too, etc. Okay. You might see an adenine with a uracil, but that's in RNA. Cool. Let's continue on. All right, you have the atomic structure. So if we really look at the nucleotides, remember the singular unit, and we go deep, deep down, we have three different things here. Okay. With a phosphate group. So if we look at so we have the chemistry representation on the left here. So you have all your different elements, all right, and or atoms, whatever you want to say. And then you have your cartoon representation, which is probably simpler to look at. Okay, so phosphate group here, that's your P. All right. As you can see, it's a phosphate group. So yes, there's a phosphate here, but it's also connected to some oxygens through some different bonds. All right, that's not important, but you need to remember the phosphate. That's the important part. All right, sugar phosphate backbone. All right, a 5-carbon pentose sugar. So pentagon, 5 points. O's means sugar. All right, glucose is a type of sugar. Galactose is a type of sugar. Lactose is a type of sugar that I can't digest very well. Okay, anything ending with O's is a type of sugar or a type of carbohydrate. So pentose, so a 5-carbon sugar. So 1, 2, 3, 4, 5, always represented with that pentagon. All right, sugar. And then you have a nitrogenous base, which will always change. It can be an A, it can be a T, it can be a C, it can be a G, and it can be a U as well. Okay? Hopefully you got all that. So again, you should be looking at the cartoon representation. But this might help too. All right, this is just looking at the same thing, but slightly differently. Okay. And again, nucleotides are found in both DNA and RNA. So, as I said, it'll just be a single strand in your RNA and a double strand in DNA. So the 5-carbon pentose sugar is either ribose or deoxyribose. All right. And what do you think at home is the difference between those two? All right. All right, that was 5 seconds. That should be more than enough time. So if it's deoxyribose, it's going to be found in DNA. If it's ribose, it's going to be found in RNA. Ribose, nucleic acid. Deoxyribose, nucleic acid. All right, simple. Ribose has an oxygen molecule attached to the second carbon. Again, not that important, but interesting to see. So what we're looking at here is ribose. Okay, so you have that oxygen molecule. If it was deoxyribose, so DNA, it would just be exactly the same, except this oxygen molecule with the green arrow would not be there. All right, and you can see arrow representing, again, which way it's going, but in this case it's got two heads because it's showing both the prime ends, so the 5' end at the top and the 3' end at the bottom. All right, if you were to draw the opposite sort of thing, all right, hold on, where's the pen? I've never done this before. Yep, that's the pen. Okay, cool. If you were to draw this the opposite way, so an antisense, an antisense nucleotide, you'd have the nucleotide being upside down. All right, so circle. How to do this? Circle, 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 red circle. I definitely didn't draw that properly. But anyway, it looks something like that. Boom, boom, boom. All right, cool. And you would have the prime ends being different. Okay, the other way. So you'd have your 3' end up here and your 5' end up here. Again, that's not the best, but that'll do. Okay. Alrighty. So if we look at the molecule on the right here, 1 carbon, which is attached to the nitrogenous base, you have this carbon here. Okay, and as you can see, we have some molecules over here, adenine, so we should know that that's a nitrogenous base straight away. And if we look at the colors of our molecules in Head Start, we should be aware that this has a few carbons in there, it has a few hydrogens in there, and the blue molecule, nitrogen, super important for building these amino acids. Oh, sorry, for building these nucleic acids, but also important for amino acids. But we have our nitrogenous base here, okay? So made of nitrogen, nitrogenous base. The name should line up there. Cool. 3rd carbon molecule, which attaches to the phosphate of the following nucleotide. So this molecule here of carbon, oxygen, and hydrogen, that will attach to the phosphate group, so the top here of the previous nucleotide. Okay? And the 5 carbon, so this one here, which attaches to the 5 carbon sugar, which attaches to the phosphate group of our nucleotide. So orange molecule, phosphate, with lots of oxygens around it, that's our phosphate group. All right, now that's quite important, especially because we can count these phosphates. So 5 being the top, 3 being the bottom. All right, and if that's flipped upside down, 3 is going to be the top, 5 is going to be the bottom. So lastly, what is this molecule here called? That's right, you definitely know what it is. Adenine and 3 phosphates. Adenosine triphosphate. Adenosine triphosphate, also known as ATP. Where have we heard of that before? All right, again, as previously mentioned, RNA uses a different nitrogenous base. Okay, so the best way to remember this one is that A and T, they have a weak bond, which will come back later, ironically. They have a weak bond. So T is prone to cheating on A. and it cheats with U, also known as uracil. Okay? Whereas C and G, their bond is much stronger, so they would never cheat on one another. Alrighty, so we have our nucleotide here. Pretty simply, you have a nitrogenous base here, and it's A, C, G, or U, can be any of them, so that has to be RNA. Okay, we can also see this by another thing here. What would it be? That's right, you guys are really good. It's that pentose sugar. Alright? As you can see, it says ribose. So that's RNA. Whereas here, deoxyribose. Alright? No oxygen attached, so it's deoxy. Deoxy. No oxygen attached. Deoxygenated ribose. Again, the names give you everything in biology. Alright. Cool. So A pairs with U, alright, if it's RNA. Whereas if it's DNA, A pairs with T. Again, C and G have a really strong bond, so they're always together no matter what. Lastly, we have the type of nucleotide structure. Again, this probably isn't the most important thing, but something to remember. And there is a little pseudonym, I guess you could say. So we have purines and pyrimidines. So purines, this is how I remember it. They're pure, so angels and goodness. So it's A and G with the double ring structure. So you can see that in the first two. So adenine, guanine, angels, goodness. Then you have your pyrimidines. Okay, so that's the bottom three. So they're singular ring structure. So what I mean is one ring, literally. Whereas these ones at the top have two rings. All right, this is more chemistry, but still something to note. So the pyrimidines, which ones are they? The pyramids were made with tools and cement, so that's T and C. Again, probably sandstone rather than cement, but anyway. Tools and cement, and then obviously wherever you have T, it's also going to be U as well. So T and U, remember, interchangeable in our nucleic acids. Awesome. Cool. And that's it for today. So that's your first class. All done and wrapped up in 17 minutes. Again, you can take notes on this. You can do what you like. I'm going to continue to do these videos throughout the year. If I get too busy, I might miss a few, but we'll see. And I'll just post these on YouTube. Send them to your friends. Send them to anyone who wants to learn some biology. And yeah, thanks for watching. Mr. D out.